This session is cross technologies, talking about omics, crossing omics, lots of omics. I know there's a lot of omics here in general. Welcome to another section on omics. We have 3 talks and a sponsor and a lightning talk, so we're gonna dive right in. First, we have Spencer Schwartz with Canopy Biosciences here as our sponsor. So thank you. Come on up. One moment of housekeeping, which you all should know. Please silence your phones. Photography is permitted. And please hold questions until the end of the sessions. And we have an app. I'm sure you've all figured that out before. Okay, awesome. Thanks, everybody.
Cool. All right. Cool. Thank you for the, there we go. Happens a lot. Thank you for the introduction. Yeah, I'm Spencer Schwartz. I represent Canopy Biosciences. Canopy sells a multiplex, a spatial biology system called the CellScape. So what I'd like to talk to you about for the next 10 minutes or so is the data that's been generated using CellScape. Before we really get into it, though, I did want to mention that we've had a couple of new tools that we've added to the CellScape ecosystem, whatever you want to call it, very recently. First is we now have whole slide imaging. This means that you can use anything that's already pre-slided. If you're working with clinical applications where they've already cut tissue, this is something that we can work with now, too. Additionally, that imaging area is 16 millimeters by 40 millimeters, so that also means that's essentially the entire working area of that histology slide. Finally, we now have validated and targeted kits. So this is essentially a way to make sure that you're starting off with these spatial biology applications on the right foot by having some markers or having some antibodies that are already kind of dialed in, say, so that you can use them right off the bat. So talking about data. So what we're looking at here is a hyperplastic tonsil. So we stain this with 15 different markers. It's 15 millimeters by 9 millimeters. But importantly, it's sampled at 5.5 pixels per micron, 182 nanometers per pixel. So this is a highly, highly dense data set. So when it comes down to it, this is actually 721 megapixels. So before we even talk about the intensity variation that we have on it, the number of markers, it's just a very big image. But it's also a very powerful image. So out of this, we were able to extract 1.8 million cells. So this is kind of where we are with spatial biology in general. We can look at millions of cells at once. We can look at whole images. We really are kind of ready to go. So what I'd like to do for the next kind of 8 minutes or so, 7 minutes and 32 seconds, is really kind of double down on this idea that we really do need to collect as high density data, as high resolution data as we possibly can. So there's another little caveat to the way we collect this data. So this is multiplexed IF. This is cyclic multiplexed IF. So essentially what we do is instead of putting all those antibodies on at once, instead we build these through sets of 5, cycles of 5. So we'd say stain for 5 antibodies, we would collect that imaging, and then we would say perhaps put that sample in the fridge, bring it out a little bit later, and continue the cycle. What's key to this, though, is you do have that ability while it's in the fridge to actually take a deep breath and decide what markers you want to sample for next. What this allows you to do is what we call hypothesis-driven assay development. So in the case of, stop touching things. So in the case of the data set that we're looking at, so this was 34 markers on an adenocarcinoma lung. But very specifically what we did was we took a look at first the spatial immune profiling kit, which is only 12 markers. We took a look at that data set and saw within it that it might have TLS structures, tertiary lymphoid structures. So instead of staining for 1,000 markers and hoping one of them sticks, we could directly stain for just the markers that would identify that particular tissue, get the data that we need out of that, and then move on to the next round of imaging. What this allows you to do is you never waste marker, you never waste resources on markers that you're not necessarily interested in just because they're part of a kit or part of an application. And otherwise, you also don't waste a bunch of reagent on maybe what could be a dud sample. Not everything stains. So this is really kind of the ultimate optimization of this sort of work, is cyclic immunofluorescence. So Again, this was 34 markers. We did this actually over a period of 2 months, and the sample is actually still in the fridge available for us to stain again. So this, I think, for this particular audience is incredibly powerful. Imagine if your users could come to you week after week, month after month, with their new experiment on our, the same machine, the same prep, and just add new markers to it and let them build these cyclic immune profiling stains. Much easier than trying to validate a 100-marker panel. So, again, let's kind of talk about, there we go, the data itself. So what I'd really like you to notice is really kind of the high contrast quality of this image and also the resolution. So here we're looking at an invasion of immune cells into a pleural space, but really what I want you to see here is that, for any of those individual markers, I don't just see if it's positive or negative, I can actually see polarization on those markers. I can really fine-tune what I'm actually seeing, and again, us imagers or people that are images, we just call this morphology. So what we're doing by sampling at a very high resolution with high contrast imaging is getting that morphology information, which is incredibly important. So there's two things that kind of go into this. The first, of course, is high sampling, so 182 nanometers per pixel. The second is we do this collection through HDR imaging. So instead of a single image with a single exposure that's going to have a limited dynamic range, we actually collect a bunch of exposures, and we collect the entire expression level of that particular marker. In doing so, we're able to show very high contrast images. We can show the whole, sorry, how about that? All right. But in doing so, not every marker requires HDR imaging, but every marker can benefit from HDR imaging. So kind of taking a look at interactions here. So if we really zoom in here, so what we're looking at here is T cells, B cells, and macrophages interacting with each other in order to eradicate these tumor cells. What I think is really cool here, going to do this? Cool. So with this B cell on the left, I can truly say that it's interacting because I can see that it's interacting. It actually does have protuberances. They're going into that T cell. I don't have to add 34 other markers in order to say this. This is buried in the morphology already. Same thing on the left. Those are 2 T cells attacking cancer cells. So I don't have to say maybe they are. I can see that they're interacting. I can see the envelope of the membrane that's surrounding those other cells. So this in itself is really very powerful, and this is really what people want out of imaging. This is what microscopists see all the time. This is what actually these deep learning algorithms are trying to encode. It's incredibly powerful stuff. We take a look at another example really quick here. So these are T cells attacking cancer again. So on the left, you see a T cell that's actually wrapped around those cancer cells. So T cells, if they're happy, if they're active, they're incredibly active. So they're swimming around the cancer cells and they're attacking them over and over again. These are live cell favorites because this kind of dance of death is just so pretty. So in contrast, though, if I take a look at those 2 T cells on the left, they're not doing much of anything, and they're also balled up. So I can see through that morphology that there's probably some sort of suppression, maybe some immunosuppression going on, without having to necessarily stain for PD-1 or PD-L1 or look at those checkpoint inhibitors. But here's what's really cool about CellScape. If that's my question, I can go back, stain that same sample for PD-L1 and PD-1, and actually verify whether that's happening or not. Final example here. I'll get out of your hair. So this is a true example where you might actually need HDR. So here we're looking at PD-1, PD-L1 on cancer versus essentially T cells attacking cancer cells again. What you'll notice is this blazing bright PD-1 and also some fairly high PD-L1. So essentially what's happening here is you can see this positive feedback loop where it's not able to actually clear those cells, so it cranks up PD-1 in order to try to do that, and it's still unable to do so. But the thing is, if we pull back some of these markers and just take a look at PD-L1 and zoom in a little bit, put it in mono, and of course, look at it in a bright room. So take my word for it here that there is actually some very dim PD-L1 in addition to this blazing bright PD-L1 that you see in the cancer cells. The thing is, PD-L1 is it's expressed on basically anything that comes out of bone marrow. So it's not necessarily whether it's binary, it has the signal or it isn't. It's rather, is that signal that we're seeing, is that outside of the endogenous expression that I would necessarily expect? That's where you need HDR imaging because we need to be able to collect on a linear scale all that data in order to measure those two one against the other. So that's an example where HDR actually does do you a specific benefit. It allows you to access to something that you wouldn't otherwise have. Kind of wrapping up here though. So, just to kind of double down here again. So can CellScape give you clustering analysis and do phenotyping? Absolutely. Can it do neighborhood analysis? Absolutely. But I'd say that's the state of the art now. What's coming in the next 2 to 3 years, and again, this is powered by AI, by deep learning, is trying to trying to decode this sort of image. So this morphology is really why people do microscopy in the first place. So this is why we need very high resolution imaging, very high contrast imaging in order to make sure that we have the data set so that when we're ready to go with these deep learning methods, we don't find out later that we undersampled or anything like that. So this is why I think that CellScape particularly is a really, really powerful tool that you could add to your course in order to really do very nice spatial biology and do it practically and do it at scale. Cool. I appreciate it. Thank you. That's I did great this time. That's my time. I appreciate it. Please visit us at the booth, and if you have any questions, I'm happy to take them there. Take care.
Thank you, Spencer. Next, we have a lightning talk. This is Kevin Mayer, who's an R&D scientist at Promega.
Okay, good afternoon, everyone. I'm just going to get started. So FFPE is an important clinical sample type. Nucleic acids can be purified from it, and downstream assays like NGS can be used to understand the genetic basis of disease. But it's challenging to work with. And I like this quote from Keith at his Omics blog. FFPE samples are the darling of the pathology world and the bane of the genomics community. And this is really because formaldehyde is extremely reactive to biological amines in the cell and creates extensive crosslinks. So before nucleic acids can really be used in downstream techniques, the crosslinks need to be reversed. And the industry standard for this in a purification workflow is a long heat incubation. So today I'm going to share work we've done at Promega R&D to employ chemical strategies to selectively and quicken this process. So we first characterized the role of the decrosslinking incubation for an FFPE DNA purification. And as you can see here, too little time, that DNA is still cross-linked. It doesn't purify well, it doesn't hybridize, it doesn't amplify by polymerase. And too long, the DNA quality is compromised and begins to degrade. So there's a sweet spot of between 4 and 8 hours, and this is temperature dependent. So we can make NGS libraries through targeted sequencing, and what we see when we do S&P analysis is that the length and temperature of this decrosslinking reaction is really driving S&P artifacts. In particular, these C to T S&Ps, which are a common artifact found in FFPE. If you're involved in NGS of FFPE, you might recognize them. And this is the result of cytosine deaminating to uracil. So there are commercial solutions for this. Uracil DNA glycosylase can be used to remove this, but it doesn't get rid of all of the deamination events, and it does reduce library complexity. So we were very motivated to reduce the necessary decrosslinking time for FFPE. So we initiated a chemical screen. So what I'm showing you here is amplifiable DNA yields. Each column or number is going to be a different compound that we put into the FFPE lysate before the decrosslinking reaction. And so we started with a wide range of compounds that have different functional groups at different concentrations and pHs. And for those that we see bumps in yields, we take those functional groups and we explore the chemical space around them and iterate until we find catalysts that provide just as much amplifiable DNA at the suboptimal decrosslinking times as like a 4-hour optimal control. So what we see is DNA yields across decrosslinking time, and when we add in these catalysts, we're essentially compressing the kinetics of this reaction 8 to 16-fold. And when we do NGS with some of these catalytic-driven reactions at low decrosslinking times, we are seeing a reduction in S&P artifacts that is quite competitive with workflows that use UDG enzyme. So with that, I just want to say thank you for your attention. Thank you to my exceptional colleagues, and reach out to any of us if you have questions about these types of technologies. Thank you.
Ooh, the timer just gave me 62 minutes, I think. So we're in for the long haul, everybody, right? Yeah, I think, so Technically, the session information says this is technically a panel, but I think the three of us that are giving talks actually have pretty disparate talks. So let's go ahead and, within each of our time boxes, feel free to ask each of us questions immediately after the talk, and then if we have extra time at the end, it would be lovely to field further questions. Yeah, and so with that, go. Okay, so set my own timer here. So hi everybody, thank you for coming. I'm Erin Weisbart. I am a computational scientist and an image analyst in the Piximi lab, in the imaging platform at the Broad Institute, and our field, our niche, is in quantitative high-content imaging. And so my pitch to you is that we're sort of working at the intersection of imaging and bioinformatics, and doing imaging omics, right? Since that's what this session is, all about omics. So I'm going to talk you through why images, why do we care about them? Why do we pick images as our omics? I'm going to tell you about some advances in software and data science and machine learning that are enabling us to get even more quantitative information out of our images. And I will touch on hopefully, the importance of public data, open source software, and cross-disciplinary collaboration, because those are three things that I care about a whole lot. Okay, why images? Well, they're really informative. First of all, they're pretty, right? It's a lot of fun to stare at these all day long. So pictures are pretty, but more than that, they can be so much more. So image data can be single cell resolution. It can show you interaction between molecules. It can show you live cell information. It can show you changes in expression level. It can show you changes in behavior. And beyond all of that, biological images can be quantitative. However, microscopy's history is mostly qualitative, right? If you think about, some of the very original observations of cells under a microscope, it is a biologist looking at a picture and describing what they see. But that's not where the field is anymore. I think we all understand that imaging can be very quantitative, and in the imaging platform at the Broad, we very much push this understanding that we can use software to turn images, which again are quantitative information. So we can take that the quantitative information in an image, extract numbers out of it, and then use those numbers to answer biological questions, to drive biology. So we have a number of softwares that we write and maintain. Hopefully anyone that works with images has at least heard of CellProfiler. It's been around for 20 years, and we are still very actively developing it, a very popular tool in imaging, but we have a number of other tools, some of which I will touch on in the talk. I will say, one other cool pitch for the imaging platform is that we have lots of software engineers and we have lots of biologists, and we all work together. And I think that's one of the things that makes us sort of uniquely able to really push forward imaging as an omics, because we have softwares writing great software that, but also biologists driving that, helping to communicate to the software engineers. This is how a biologist thinks about the problem. This is what the biologist actually cares about in the software. And so together, we build cool tools. So what is this intersection of bioinformatics and imaging that I'm talking about? Well, if you think about it, digital images really are just arrays. This is a really simple example where these pixels are scaled by their intensity, and sort of intuitively, you can see these dark pixels on the outside, are the smaller numbers in the array, and the brighter pixels on the inside are the larger numbers in the array. And here is the array that describes this image. So this image of this dot is quantitative. That is a digital image right there. You can easily expand upon that thought and say, okay, if we have a much more complex image, we have a more complex array of numbers, right? I have a much bigger X and Y, much bigger intensity range. You can have layers upon layers of information, right, that are all, all your different channels. But at their core, we're just looking at arrays of numbers, and with arrays of numbers, you can do math. And math is quantitative. Hopefully I don't have to convince you of that one. So as I mentioned, we develop and maintain CellProfiler, which is an open source software for image analysis. We think it's pretty cool. It is free and open source, available for Windows, Mac, and Linux. Independent analyses, so I mean, obviously I'm biased because I work on it, but independent people have also ranked it as very flexible and usable. And it is used by many of the top pharma companies, cited in many papers per year. And these are just some of the leaders in our team that have, have been team leads on CellProfiler over the years. So it has a simple interface that, I don't have the time to go into great depth, unfortunately, because I can also, if I had it adjusted itself, it says 15 minutes now. I don't actually get the full 62 to really convince you about how awesome it is, but you have a simple GUI where along the side here, you can drag and drop in modules, and every module you can then tune within every module has its own set of settings that you can tune. And so any biologist, you don't have to code at all to use CellProfiler. Any biologist can take whatever modules they want, drag and drop them into this workflow pipeline here, tune them however they want, and use them to analyze their images. So we have currently 94 modules that collectively have 930 settings. So lots and lots and lots of things that you can do. But I really want to highlight our total library is 120,000 lines of code. 37,000 lines of those of which are documentation. So again, we are biologists, and we very much want to help other biologists to be able to use quantitative software methods. And so we very much value both documenting and teaching how to do what we do. And this, lines of documentation code is just one example. So one thing you can do, and which is usually the start of a workflow for many of the sorts of assays that we run in our lab, is to identify objects. There are many, many different methods you can do this. Again, many different modules that you can pick and tune the settings to get the best segmentations. Here I have an example of cells that have a cytoplasmic stain in green and a nuclear stain in pink here, and I'm able to identify cell and nuclear objects. So then once you have objects, you can make measurements, right? Because you can know what the numbers in the array are within those objects. So we can do intensity measurements that will tell you many things, including the total intensity within that object, the mean, median, and standard deviation of intensities within that object. You can go by quartiles, you can do min and max. I mean, there's all sorts of math, right? You can do very simple math, you can do very complex math, you can do math because you have an array. We can do sort of more descriptive things using math. So we can look at intensity distributions, for example, where we can say, okay, not only am I interested about the overall intensity within an object, I want to know more about where is that intensity. Is it shifted to one side of the cell? Is it more concentrated at the center of the cell? We can use math to describe this in a very quantitative fashion. We can measure object size and shape. You can look at, area and perimeter and eccentricity and axis length and solidity and all sorts of mathematical descriptors of size and shape. We can look at object co-localization, right? So if we have more than one stain, more than one channel that we're imaging, we can ask how well do the intensities in one channel correlate with intensities in another channel, like within our objects. To do that, here's just two examples, one of which don't correlate very well, and you can see intensity in one channel going one way and intensity in another sort of off the other axis, versus here, where they're highly correlated, you see correlation. There's all sorts of ways that you can look at this, and you can do this, across any of the objects that you might have, or your whole image. You don't have to segment objects. There's lots of interest in object segmentation. You can do whole image measurements, right? You don't even have to have an object in here. We can do complicated math that I don't even really fully understand, right? Zernike polynomials. Maybe there's somebody in here that really understands them. I can tell you they are a descriptor of shapes of objects, and you can use many Zernike polynomials together to pretty accurately reconstruct shapes. But this math goes beyond my head. But the cool thing is I don't actually have to understand Zernike polynomials. I can know that they are a way of describing object shape. And I can use them still as features to quantitatively describe my data. So you can have very interpretable features. You can have features that are less easy for a human to interpret, but are still very descriptive of your data, and therefore very useful. We have other things such as texture and granularity, other examples of categories of math you can do that is not terribly human interpretable, but very nicely provides features that explain the differences in your images. So all of this combines into the field of image-based or morphological profiling, which is basically imaging omics, which is one thing that our lab excels in. And so, here in this example, we have, you can take healthy or diseased patient, or patient cell lines. You can perturb them with drugs or genetic perturbations. We tend to often work at scale, so you can do high throughput things at scale. You can segment them, stain lots of things. Here we are finding nuclei and cell objects, and then making lots and lots of measurements using all of the measurements I just described. And when we do that, we have profiles. So you may be very familiar with looking at sort of omics data, right? So in this parallel, each line I have is a single cell where I have a robust description. And rather than having gene expression, for example, a different gene in my, along my columns, in this instance I have image features. And so you can get single cell data that is fully quantitative and a image-based profile that describes your quantitative image. And then you can do biology and ask questions. The assay that we work with the most is the cell painting assay, which uses 6 stains across 5 channels that stain 8 organelles or cellular compartments. It is very, very robust. You can do all sorts of things with the cell painting assay. And it's about half to a quarter the cost of mRNA profiling using the L1000 assay, for example. So, these are all stains that are very easy to stain and don't require any special imaging. You can do them sort of on any standard wide field fluorescent microscope. You can run this assay. So, in summary, for cell painting, we identify nuclei, cells, and cytoplasm, and then we measure everything. That's our assay. So the cell painting assay itself has been used for many, many things, and unfortunately, I can't go into all the things, but I will tell you, we've shown it is great for predicting small molecule activity, characterizing gene alleles, predicting cellular drug resistance, distinguishing between disease states. You can predict the outcome of other assays based on the morphological profile. And we know that it complements nicely other profiling techniques. So there is information contained in cell painting that both overlaps and is distinct from mRNA profiling or protein profiling, for example. So it is, that there is unique information within morphological profiling that you don't get from other omics. So hopefully I've now convinced you what we do is cool. It has many applications, and I'm just going to go into now a few examples. Again, don't have the time to tell you all the things I want to tell you, but one thing that we are working on, sort of crossing omics, since that's the theme of this session, is using bioinformatics tools that have been developed in other omics, for example, for RNA-seq, tools that have been used for data set alignment or for batch correction. We are actively exploring how that can be applied to morphological profiling data. That's a cool thing that, since again this is just quantitative numbers, so we can take tools across any bioinformatics tool developed anywhere, and we can then apply it, expand upon those methods, use them for our morphological profiling, and hopefully even develop new methods that can then be taken elsewhere as well. We are also pushing forward development of what you can actually do with the cell painting assay. So we have created in a giant partnership, but if you want to talk about this, I've been working on this for 5 years now, so come talk to me afterwards, where we can do cell painting in a pooled format. So instead of needing to do it in an arrayed format where you have a single perturbation in every well, we can do this in a pooled format and then use sequencing by synthesis, where you have like optical barcode calls, where we can actually then assign a perturbation to every cell individually within a large pooled format. So you can dramatically scale your ability to scale up perturbations. So we can do a whole genome knockout experiment in six well plates at a fraction of the cost of many other whole genome perturbation type studies. So as I wrap up, I want to say, how can we bring quantitative bioimaging to everyone? Because clearly it's important, it's good, and how do we proselytize this even further? So I think one really important thing is fair data. I have a lot to say on this and not enough time, but hopefully everybody is thinking, I have heard fair data being talked about throughout the conference already, so hopefully everybody, no matter what omics you're in, is thinking about how you can share your data in a way that other people can reuse it. And that democratizes such things so that people who do not have the resources to generate the data, are still able to mine it for many, many things that I'm sure whoever has generated it has not fully delved into. One example of that, so I'm lead curator, I guess, on the cell painting gallery in Rhoda, where we have now almost 700 terabytes of cell painting data and corresponding morphological profiles that are freely available for anyone to access. And one example of that data set, if you're in the if you've heard of cell painting, then perhaps you've heard of JUMP, where we had a project across many, many sites, many companies participated in this to make a massive data set of over 100,000 small molecules, all profiled using cell painting, and it's all public and available for anyone to mine. Out of time, so I will also say I love next generation file formats, and I think I'm happy to talk about file formats, which is probably the least sexy slide I have in my whole talk, but if you care about file formats, please talk to me also. And finally, we also write software that makes it easy for others to do things at scale. So this is one example of our softwares where we are helping to simplify the process of distributing compute across AWS so that anybody can more easily do their experiments at scale. Deep learning. Also a recurrent theme seems to be. Everybody's very excited about where it's going and what it's doing. If you want to hear more about AI and image analysis workflows, there's a great session tomorrow at 2 o'clock, so please make sure to go check that out. And I think with that, the final thing I'm going to say, because I'm out of time, is one example of one other software that we are very actively developing that is deep learning on images entirely in your browser with no code. So you can actually go to piximi.app right now. You can drop in your own images, and you can do deep learning on your images in a browser on your laptop with absolutely no need to code anything. And so we're really excited about that. And again, lots to say and not enough time. So I will end by thanking the members of the Piximi Lab and the Carpenter Singh Lab. We collaborate very closely in the imaging platform, and thank you all for your attention. So I think with that, I used up my question time talking, and so I'm happy to field questions at the end, but I will invite up Kaylene now for her talk. So Kaylene Simpson is at the Peter MacCallum Cancer Center, and she came here from Melbourne just to join us today, so thank you for the long distance flight, Kaylene.
Shrink down a little bit. Well, thank you for inviting me for the long haul, because it's been a fantastic meeting. Really enjoyed it my first time here. Work out whether I need glasses or no glasses. So I head the Victorian Centre for Functional Genomics, which is a platform technology at the Peter MacCallum Cancer Center in Melbourne in Australia. But we're a nationwide outward-facing facility as well, so it's not just the people in the Peter Mac that work with us. And as you can see on the left there, we're a little bit of everything to everyone. We started as an RNAi screening facility, we've moved through CRISPR, we do a lot of drug screening, and I'm going to show you today about what we've been doing in automating our organoid platform as well. Our model is for people to come in and work with us. We do fee for service, but typically we prefer to bring that next generation of scientists in, train them, work side by side with us, so the team will always be there to support them, and we drive all the data analysis. So our model here is to drive innovation, drive discovery, and have impact. So today I'm just going to whip through, because I should have talked to Erin before, because we have a few similar slides, the idea about discovery-based screening, and then introduce you a new technology that we've developed called Mass-Spec, and then just touch on how we've been working with organoids in a 3D automated setting, and how that's driving something towards personalised medicine. So discovery, high-throughput screening, the way I like to describe this is going from a lot down to a little. This inverted triangle that you can see here, where you've got a primary screening strategy where you might have thousands of something, CRISPRs or compounds, for example, and you are using iterative data analyses to drive yourself through secondary screening, tertiary screening, and developing a high confidence list. The important thing about this is that it's unbiased to a certain extent. You're always going to have to overlay something that you know as you get down the pointy end, but that it's there to be using the best model for your biology. I think the days of just using a U2RS to represent breast cancer are over. That was what we did many years ago. Oh, we're back. And so just on the right here is sort of more use cases where the thousands of screening agents is very high throughput. We can do that in adherent or suspension cells, but typically we're seeing screens that are multiple cell lines these days. Synthetic interaction screens are very popular, host pathogen screens as well. When we're down here at the validation point, that could also be the primary point of a starting screen where it's a really complicated, more difficult and expensive screen, or just the point where you're doing lower throughput and the smaller numbers of targets for validation. At the bottom here, tertiary screens are usually orthogonal assays, more sophisticated assays where you're working towards getting a high confidence target list. And in practice, it looks like this. We're working usually in a 384 well plate format. We deliver our agents into every well of these plates. We perturb for somewhere in the order of 3 to 7 days, and then we acquire our outcomes, whether they be imaging, plate reader, or flow cytometry typically. And then the data analysis is where it really gets fun. We don't like to see any data going out without us having our eyes over it, so we will always report on the QC. Most people come and say, I want to see my hits, and I just say, I don't really care about your hits. Until I know your data is good, we don't worry about anything else. So we'll go through all the fundamental QC steps. We'll usually offer a basic to an advanced analysis package. But on top of that, we do a lot of machine-based innovation, machine learning innovation, but everything for everyone is fully customised, so they get the best outcomes they can. Erin's already introduced the idea of cell painting, so I don't need to say any more about that, other than to say that we don't use it every time. It is a little expensive in Australian dollars. So sometimes we do the poor man's cell painting, and we might just have 1 or 2 channels. Here we've just got DAPI and a phalloidin channel. Phalloidin discriminates biology beautifully, and what I'm a big advocate for is adding in your target X, staining for something else that's relevant to your biology, so that it may be coupled with the alterations in cell machinery, but it's very specific to the question that you're asking. And so our pipeline, as the way we describe it, very much as Erin's, is taking your images here and then feeding it through the CellProfiler pipeline. We will always report on viability, toxicity, and cell cycle as read. That will be done for every single screen. But our feature extractions, we are using that in a 2D setting and also in a 3D setting. We actually co-opted parts of the CellProfiler pipeline to develop our own 3D pipeline. So we extract our cell features, we use phenotypic clustering and feature reduction to come up with feature fingerprints, and you can see here, these aren't exact data, but they're the stylized version to show you we could have different types of fingerprints and very different types of cellular images. And then we get to the end game, where we're going to integrate other screens, GWAS, clinical data, publicly available information, but we're still left with that idea that we've got biological pathways, but how do we know which is the winner? Which cluster of targets might we want to follow up? And that's, how do we determine that biological relevance? And so here's where we introduce Mass-Spec. We've called it Mass-Spec, titled for multiplex analysis of cells. There are some other papers calling it drug-seq and plate-seq. This is our version of that, largely because we've had talks on findable and reusable, and we couldn't get some of those methods to work. So we made our own. But it's high-throughput transcriptomics. It's cost-effective, 384-well-based format transcriptomics. And the feed-in can be anything. It dovetails through all of the screening modalities we have, and then other people come because they just want to miniaturize their assays and just do a faster, cheaper version. So here's where we perturb our cells in either a 2D or a 3D setting, and we lyse those in situ in the plate. We then reverse transcribe in that plate with a unique barcode for every well, purify, PCR, amplify, and tagment, and then we pull the whole lot together and we do one sequencing run. And then the deconvolution magic happens with the data analysts. I'll just show you that we can do this for cells in 3D, and this is really important if we're taking primary patient material and we want to know that it's repeating its biological profile over the time that we've got it in culture, but it took us quite a bit of work to work out how to do that. So we have to depolymerize the matrigel. If you leave that too long or it's too aggressive, you will most certainly stress the cells out, and that's borne out very quickly in the sequencing. But we've got a method for that now. We can drop them down to the bottom, we can wash them off, and we can get them out. And so from there, we actually have quite a few cells usually, so we can do this RNA isolation for the Mass-Spec, but we can also do bulk or single-cell proteomics, and we're even getting greedy enough to take the supernatant and try some cytokine profiling or some metabolomic profiling. This is just the early work that we did to confirm that this method was going to work. We just took some ovarian cultured cells. We had a number of different cell densities. We had a number of different treatments where we had media and DMSO, and then we had some tox-killing agents. And you can see here we're probably bordering on the threshold of not great input at 450-odd seeding cells. So we've only seeded them and then had very short time points, so not a lot of cell proliferation's gone on. But once we hit slightly larger cell numbers, there's a great consistency in the number of genes detected, so we felt quite comfortable that the method was working in a 2D setting. So then we progressed to a 3D setting, and this is just using MCF7 cells in 3D. And we do a small mechanism of action library screen, just 69 compounds over a dose range, single time point. And again, the total mat reads and the total genes detected didn't vary at all based on any of these conditions, so we're again satisfied that the method was working well. So we took that a little bit further and we went and analysed the data from that little mechanism of action screen in the 3D cells, and we picked up on this compound called luminespib, which is an HSP90 inhibitor. And for the low, medium and high doses, we got targets going up, targets going down, and of course no target change. So then when we looked at the top 10 differentially expressed genes in each one of those, you can see the low, medium, high. The overlap was 6, and of those, 5 of those were HSP target genes. So we thought this is looking good, the method may actually be working. But just to be sure, we took 2 other agents that were toxicity targets, mitomycin C, oligomycin A, and when we looked at the top 10 differentially expressed genes in those, we got a 0 overlap. So we're really happy that the method looked like it was actually going to work. And then it gets a bit more fun, because here's when we start to integrate our imaging omics. So I'm glad you coined that term, because we're calling this imaging transcriptomics when we play in this space. So the same screen that we set up, we've got our transcriptional information and we've got our biological information. So here the lead was, if we're going to do this multiplex analysis in 3D, can we cluster, and what do we cluster together? So we clustered these two compounds with this same profile. This is a very stylized diagram here of what happens next. We were looking at pathway analysis, but actually we picked out NF-kappa B as differentially expressed target, which are the targets of this compound here. So again, all little steps, known agents, but still giving us something that says this method's going to work. So how do we apply it in real terms? This is a really big screen. It's been led by Dane Cheesley and Kathleen Pishus at Peter Mac, who collaborate with Mark Carey at Vancouver. Mark's the guy who has all of the primary patient cell lines for low-grade serous ovarian carcinoma. So this is a really dreadful disease. There's not a lot of treatment for this disease. So the whole premise behind this was to find new compounds to treat this disease. So we set off to screen known agents still, FDA library, kinase inhibitor library, epigenetics targets, but following through that lot to a little inverted triangle, we've validated, we've done this over 13 cell lines. So 12 of these patient lines and 1 normal cell line. We've characterized the lines inside out, growth kinetics, small pilot screens. We determined the screening regime that we would use, set that all up and ran through with just a DAPI stain, cell mask stain and a phalloidin stain. We also imaged at 10 and 20x because, as you talked before, getting more is better from the beginning, so if we ever need to go back, we've got it all in-house there. And then we went through a series of customised analysis pipelines and integrated that with all the molecular and clinical data this team already had on these cell lines as well. So all of that brought us down to a hit ID of 79 high-confidence compounds. And then the strategy after that was to embark on another screening campaign where we then sought to find which of these targets would interact with standard of care treatment. So the idea being to augment whatever's in the clinic at this point in time, and then to go and study those using Mass-Spec. So it can be a little contentious to filter out against a control cell line when you've only got one control cell line and so many cancer cell lines, but we did it for the purpose of just seeing if it would be informative to us. And so you can see that the black markers here are those that have been the hits and their target pathways that have been filtered out that have no effect in the control cell line, and that they, by and large, are enriched for compounds that target the MEK, EGFR, and the mTOR pathway. So the synergy screening was a tour de force, actually. We have done a 6x6 dosing grid. We've got 6 anchors, or the standard of care treatment, and we've done them in combination of the 79 hits across 4 cell lines in the first instance. So it's 180 plates, I think, per cell line as we've gone through it. But you can see this little graph on the corner shows that we've got quite an enrichment using 2 different synergy software tools. There's actually 4 that we use, and we're trying to get a little better statistical hold on which is the best one to use. So we still will rank a couple against each other. And these synergy plots over here show the different EGFR inhibitors at the bottom and the interactions we've got with just a couple of those target hits. And they're all coming up really robustly. So we then proceeded to drive Mass-Spec first experiment where we were just taking different EGFR and MEK inhibitors, 1 cell line, 1 concentration, 1 time point. The time points can be the challenge to work out when is the best time to take them. So we're still sort of getting a sweet spot, but we did some primary optimization for this. And this graph really is just here to show you that this does work. We've got quite a high overlap of compounds and targeting pathways that we'll be able to follow up down the track. Some independent ones, but this is the study that we'll take forwards to understand this in this context. And so in our own lab, we've been always wondering whether or not you could drug and do a CRISPR screen and coalesce the 2 targets based on imaging phenotype. I think it's probably more difficult than that in the sense that you've got a knockout or a suppression, and you've got a drug, so you're never going to have a complete one-to-one there. But we have set this experiment up where we've used that same mechanism of action compound library. We have then taken 70 targets that are representing that mechanism of action library in RNAi and in single guide CRISPR using the synthetic guides from Horizon. We've done this in spheroids, and we've done this in a 2D monolayer, and we've generated this with cell painting, multiple time points, multiple drugs, multiple imaging levels, the whole shebang. And we've done the whole lot with Mass-Spec. And now we're using this as our data set to start to understand the nuances around developing a pipeline for transcriptional profiling, developing a way to integrate the image outcomes, and whether or not truly there will be any representation across one modality to the other. So it's a work in progress right now, the transcript data, the phenotypic data, and then the cell viability data as well. So I'll leave Max-Spec and just talk to you really quickly just about the rest of the innovation we're doing in the 3D pipeline. So we own a raster and bioprinter that allows you to embed your cells in customised hydrogels, but we also own a Janus G3, and a couple of years ago the engineer in Melbourne came by to help us embed our cells, develop a protocol to embed cells in matrigel using the Janus. That has gone fabulously well, and actually PE, now Revity, wrote a white paper on how to make this methodology work. So no one needs to buy a very fancy machine if you can do this on a PE system. But coupled with that, we decided that fluorescent imaging would give you one and only one endpoint. And so we have developed a bright field imaging pipeline just using our citation plate reader imager, where we can image every single day, deliver our drugs, do a final read in bright field, and then add a fluorescence endpoint if we're interested in that. And that lets us get all of the information we need about when's the right time to actually dose or not dose, and around heterogeneity. So you can see on the top here that bright field images with organoids are still pretty ugly and very diverse. That heterogeneity in tumours is real. If you ever see one image that it's just beautiful and round and every one of them looks like that, it'll be a cell line of some sort. It's not like that in patient data. So we get this bright field image, we have to edge transform it, so we flip it so that we can actually pick them out. The brighter ones are easier to pick out. We'll then paint them with using a CellProfiler strategy. Then we've got to filter out a bit of the rubbish that's in the background of that. You will get that because you're in this matrigel setting. But it's been really powerful for us. We can now use this pipeline for all these different applications. We've done this in a huge range of PDX and PDOs throughout our institute. And it really looks like this. Using a prostate PDX, we've been able to measure growth over time. The important thing about that is that people wait till their organoids look nice and big and juicy and ready to go, and that's the wrong time to take them because they're no longer able to take up the drugs. So you really want to be hitting it down here when you're in that growth phase so that you can dose and leave for as long as you need to. So here's where we started our treatment, and you can see that they get very dark looking as they're dying, so it's easy visual readout in a black and white there. And then we can start to score for size, invasive type of features, tumour heterogeneity is certainly one as well. So that's just a quick snapshot of just the utility of doing something in bright field. We did develop a fixing and staining methodology, and we can add in the cell painting stains, but we like to offer everybody a bit of an a la carte choice when they're doing 3D. So we've got very basic screening where we're just using bright field, and we can screen as much as people like. We've done at most 1,000 compounds in 3 doses over 8 cell lines, 8 patient lines. It's sort of been the largest scale screen that we've done in organoids so far. We can do population heterogeneity, we can do time course tracking, then we can start to get a bit more sophisticated with our multiplexed fixed endpoints for phenotypic clustering, right up to where we're starting to do single cell analyses off these multiplex stained images as well, and getting heterogeneity within the organoid and then within the population as well. So we'll always have a trade-off between throughput and complexity, but it really depends on what that end user wants, and if we're getting close to that personalised medicine strategy, it might be really important for us to start playing down this end. If we could do a spatial in an organoid one day, that would be amazing. So I just want to finish with what we're doing in a precision medicine manner, and we've been very fortunate to be working with a fantastic clinician who's doing a PhD. So she's in there operating on a patient who then comes upstairs with her material from that colorectal patient. She's taking their metastases. She can get at the viable cells, and then we embark on a small drug scheme. So we're only taking the 3 standard of care agents for colorectal patients. We've got lots of replicates, big dose points, and we're just looking at death. But we're using our image-based classification that we just showed you and working out whether or not these patients would be sensitive to drug treatment. The big thing was that we could generate this for every single patient, but how do we have a benchmark and how do we make one patient relative to another? We started collecting the cohort mean response over all of those dose responses and replicates, and then Susie in my lab worked out a way to create the dose curve average, and we keep building on that every time we do a new patient, and then we worked out how to overlay that clinically relevant concentration range. So with that information in hand, we now have a clinician report that we can go back to them with, where we can look at the average of all of the patients, the dose range, and what that new patient's response is going to be based on image-based machine learning. And then we can draw them this lovely plot here that says, here's your probability that this patient is going to be sensitive to that drug. This has actually formed the basis of a new clinical trial at Peter Mac. One of the big things our institute does is a lot of clinical trials. So we're putting that through next year on whether or not we can be really predictive. Prospectively now, we're going back to all those patients' responses and building that into our model as well. And Erin, I have 9 seconds left. I'll finish with my team. I shouldn't forget them. We've got a big team. Half of us are doing data analysis these days. The other half are doing screen support services. So thanks for your time.
Yeah, thank you so much, Kaylene. We will, again, Kaylene also used up all her time, so we'll leave some, room for, find either of us with questions at the end, or if we have some extra time after our final speaker, Azi Hadadianpour, is at the Fred Hutch Cancer Research Institute in Seattle. There you go.
Thank you so much. Hi, everyone. Thank you so much for being here. So today's talk is about a comparative analysis of spatial platforms, Xenium versus Cosmics on renal cell carcinoma. I'm Azi. I work at Fred Hutch Innovation Lab as a data scientist. And I will start my talk with an outline, just giving you an introduction who we are. And then I'm going to show you some of the workflows and the sensitivity and specificity comparison that we did, and also then going to the clustering and the cell type identification, and finally ending with alternative cell segmentation methods that we have developed and the conclusion. So I'm part of Fred Hutch Innovation Lab, which is a group inside the Fred Hutchinson Cancer Center. What we do is actually try new technologies and benchmark them, and we try to tell the PIs in the labs, based on their biological questions, what would be the best approach for them and the best technology for them. So for today's talk, we had kidney samples from renal cell carcinoma, and we used Cosmics 1000-plex platform, and also the Xenium multi-tissue and cancer platform, which contains 377 genes. And we ran these kidney samples that were sequentially cut and run them through them, and we wanted to see the performance of them and how they differ. Just to give you a brief overview how these platforms work, both of these are in situ hybridization, and the thing that is different is actually the way the probes bind. So they have different types of probes, and for example, Xenium has the circulatory amplification, but instead Cosmics has the branch type format of the probes. But all in all, the way that it works is binding of the probes to the genes of interest, and then it's going to have a secondary probe that binds to it, and then you will get an image of the color, and then you have a bleaching. After the imaging, you have the bleaching, and the second round of the secondary probe comes, And then you have like this code of the colors, like red, red, green is this gene, and then red, red, red is another gene. And then that would be decoded, and you will get the data. So the way that they are different, for example, for the cosmics, the data goes into their Atomics, which is a cloud-based platform, and you can go there, and if you don't have a computational person, you can get like the UMaps from there, and like all the analysis is done there. For the Xenium, on the other hand, you have all the analysis also done on the instrument, and you can get the raw data as well. So both of them do the analysis for you, which is nice, but our experience with getting the raw data for the Xenium was easier than the Cosmics, because for the Cosmics, you have to get it to your AWS storage and then bring it down. And it was really slow. The process was slow for the transfer, but I know that they are improving on that. So as far as our analysis plan, so it is a benchmarking, so we are just doing a descriptive study and just like see how they perform. So what we did was like starting from the instrument itself, you get the imaging data, and it goes through the decoding, as I explained, and then it has a cell segmentation on board, and these are the transcripts. So you see which transcript is inside which cell, and then you can do the cell assignment and see what cells they are. They do the clustering, and they give you the count matrix of the transcriptomics. So what I want to tell you here is that you always have the option to get the decoding data and go with your other alternative cell segmentation methods. There are a lot of them out there that are well-known, like Baser and CellPose. There is another one, ProSeg, that we developed internally at Fred Hutch that I will talk about. For the sake of these slides, the very first slides, I'm actually going with the segmentation that comes from the instrument, each one of them, just to see how their segmentation also performs. And we do some QC and look at the sensitivity and specificity, and then clustering, cell type identification, and looking at the markers that are expressed differentially. So starting at the sensitivity, you can see that looking at the number of UMIs and number of genes, we definitely see much more UMI and genes in the Cosmics compared to the Xenium. However, we have to keep in mind for the Cosmics, we are looking at 950 genes, but for the Xenium, we are only looking at 377 genes. So it kind of makes sense to get more transcripts in Cosmics, but I also want to bring your attention to the Cosmics Kidney 2. So we had 2 sections, 2 samples. So Kidney 1 is working fine in both of them, but we see that Cosmics is not performing really well on our Kidney 2, which wasn't something wrong about our sample or anything. It was just like 2 samples from 2 different patients. But Xenium is actually working more consistently. So just to get a better idea of the sensitivity, we have to actually look at the genes that are shared between them. And interestingly, there is only 119 genes that are shared between these 2 platforms. So when we look at these genes that are shared, we see that the number of transcripts, now Xenium is actually doing better. So the number of transcripts, which is a measure of sensitivity, is actually better for the Xenium compared to the Cosmics. And again, we see that the Kidney 2 is not performing well for the Cosmics at all. So sensitivity is just one part of the question. The other thing that we want to see is that how specific these are. So in order to look at the specificity metrics, so I looked at different things. The very first one was like looking at the percentage of the total transcripts that are detecting genes. You might ask like what other things they might detect. So for both of these platforms, you always have 2 controls. The very first one is false codes. As I said, you have the decoding from those color codes to the name of the transcript. So they always include these false codes, which are supposed not to translate to any type of genes. So you have to have them there, and you shouldn't get any hit for those. And it is included in both in Cosmics and the Xenium. And another thing that you have is negative probes. So negative probes are there experimentally, and they are not supposed to bind to anything in your human sample. So they are just there to see how specific their probes are. So the very first thing that we looked at was like the number of transcripts that are detecting actual genes. For the Cosmics, it was 97% and 94%. And then for the Xenium, you see both of them are above 99.9%. So they have a higher level of specificity in that regard. but also looking at the false discovery rate, which I have the formula here for you if you forgot. So for the false discovery rate, I compared it for the both false codes and also for the negative probes. As you can see, again, we see a better specificity and lower level of false discovery rate in the xenium compared to the cosmics. And where it is more drastically, evident is actually for the false codes. So I believe there is, a lot of the error or specificity, decline that is coming is on the decoding part of it. Now moving on to the UMAPs and the cell types that we are detecting. So what we are seeing is that with both of the cosmics and the xenium, we were able to pick up the main cell types. And you can also see it in this bar plot that the proportion is also similar, which you expect because they are like sections that are next to each other. And they are actually, positioned in a way like that they should be similar. And you can see, like, for example, the structure of the cells are almost similar. And so in terms of the cell type identification, both of them did a good job. And also looking at the heat map of the markers, and we want to see that how good the cell types that we detected. I mean, do we have really good markers for them to justify this annotation? And we were able to see that, yeah, they have really good markers included. But I have to mention, like, the markers that are included, they are kind of different in the xenium and the cosmic. So their gene panel design is totally different. Like the genes that they have included, as I said, like only 120 of them are shared. So they are completely different. Just to dive a little bit deeper, we wanted to see that which one of them is more accurate in picking up T-cell subtypes, because everybody are excited about T-cells in the tumor microenvironment. So we were able to see, the majority of the cell types that we care about, like the cytotoxic T-cells, the naive T-cells, CD8+, and all of those. So we were able to pick them up in both of them. The only part that Cosmics was falling behind was in picking up the regulatory T-cells. Even though I saw that they have the FOXP3 and some markers for it, the expression was not probably optimized, so we were not able to pick up the regulatory T-cells. But yeah, for the xenium, we were able to pick them up. So yeah, just going through some of the things that we experienced for the cosmics, even though it is a whole tissue imaging, you can appreciate that these are like the FOVs that are next to each other, and these FOVs are not stitched. So you have like the imaging and the information coming from each FOV, and then you are kind of losing the information on the, let me show you here, sorry. You are losing the information in the border of these FOVs, as you can see, based on the number of count of the RNA. And we saw this in both of our kidney samples. But for the xenium, the imaging and the data acquisition is more, across everywhere. So you are not losing the information on the borders. Another thing that we noticed was in terms of the panel design. I have to mention, like, the panels are also developing, like they are adding genes, they're removing some of the genes, so they are like still developing. But at the time, what we saw was that in the panel that we used, Cosmics includes some genes that are highly expressed and they do not have differentiation value. As you can see here, they are expressed almost in all of the cells, and they are like highly expressed, so they can mask the expression of the other genes, especially because it's image-based, so you might get the optical crowding. And when I looked in the xenium data, I wanted to bring the xenium data also side by side. None of these genes were included there, so they probably like figured out they don't have the value for the cell type identification, so they removed them. So up to here, everything that I talked about was on one of our kidney data, and we were only looking at the cell segmentation that comes out of the machine itself. We knew that a cell segmentation is a really important part of it. So imagine that you have a cell that you get half of the transcripts from the T cell and half of the transcripts from a tumor and you call it a cell. So that would be those unknown clusters that you will end up with. And it is a really common issue because T cells are infiltrated into tumor cells. And if you don't have a good cell segmentation in place, your data all the time, you're not sure about, annotating your clusters. One thing that we saw was that none of the segmentation were ideal in our opinion. And we believe that they are also improving their cell segmentation method. As you can see, for example, Xenium is now having you to use a dye that does the membrane staining. So at the time it wasn't available. So they know that they can still improve their cell segmentation. But what we did was developing an algorithm internally. So this work was done in the Fred Hutch. So it is called ProSeg. If you're familiar with the Baser, which is a probabilistic method for cell segmentation, ProSeg is similar to Baser, but it actually improves two main issues that Baser has. The very first one is that Baser takes forever to run, and it doesn't scale properly. If you have in the level of millions of cells, it just takes forever. The other thing is that Baser is prone to over-segmentation. For example, you can even appreciate here, there are some areas that you see that there is no nucleus, but there is a bunch of transcripts, so it would call it a cell. And we know it's not a cell. And if you look at this graph here, you can see that for the Baser, you have a lot of cells that have such a low number of UMIs, and we believe those are because of the over-segmentation. But our platform, the ProSeg and 10x, are doing a better job in terms of not over-segmenting. So the paper for this one is going to come out soon, so you can look at there. So yeah, and the other thing is that now we wanted to see if we use our own cell segmentation, is it improving the data or not? And we can see that for the Xenium, when you do a better cell segmentation, you can see that the clusters are differentiated much better. This was the UMAP that we had before, and you can see that now they are much more differentiated. And for the Cosmics, it improved it a little bit, but not quite a lot. And we believe it's because there are some other factors contributing to it. For example, if you look, there are two clusters that are marked as unknown, and I will tell you why they are unknown in the next slide. So if you look at the genes that are overexpressed in these two clusters, these are mostly the system control genes, which are those false codes that I talked about. So false codes, it's really important that you don't have those false codes and at high level of expression, because it shows that there is something not going properly during the decoding. So it will always bring this question, is my signal real or not? Because I also want to bring your attention to this cluster, which is the B cells, but it also overexpressed the system control 14. So apparently whatever code sequence they put there, it was similar to what they have for the IgG genes or bank genes and all of those. So up to here, I only talked about one of our kidney data, which was a good performing one across both of the platforms. But we also had another kidney data that I showed you in the beginning. The number of UMIs were low in the Cosmics. So this is our other kidney data. And as you can see for this one, in the Cosmics, we had a bunch of cells that were unknown. And I mean, you couldn't annotate it. It was they were expressing low level of UMI, and there were no differentially expressed genes among them. But for the Xenium one, it was still performing well and picking up the main cell types that we expected. Another thing that we noticed was for these kidney data, so you always want to see your T cell markers first. So when we look at the T cell markers, we saw that they were almost absent in the Cosmics. So it definitely wasn't the best work of the Cosmics on this sample, but Xenium was doing much more consistent and robust, and we were able to see the T cell markers in the cluster that we annotated as T cell. So to conclude, I would like to say that you have to consider the setup and the runtime differences for these platforms when you are planning your experiment. Each platform, again, I have to mention that when they run okay, you know, when it's working well. Both of them were able to identify the major cell types. We saw that the Cosmics had more genes in it. It has 950 genes, so if it is more probable that your gene of interest is inside that than like 377 gene. But I have to mention that the Xenium offers the possibility to add the custom gene on top of the panel. Cosmics has that too, so you always have the option of add-on genes. Another thing that we noticed, and I mentioned, is that the high level of noise in the Cosmics makes the data interpretation somehow harder, and it might obscure those low-expressing genes. And finally, as I said, there are some high-expressor genes included in the Cosmics. That's not ideal, and also the FOV stitching was a real issue for us because we don't want to lose the data on the borders. And finally, because we looked at the 2 kidney data, we saw that the Xenium was doing good in both of them, but Cosmics failed almost in one of them, so we believe that the Xenium was offering a better and robust data for our case. And with that, I would like to acknowledge and thank everyone who were involved in this project. And we are the field lab, but we actually work closely with everyone in the Fred Hutch, and these are us here. And with that, I have 1 minute for questions, and then we are at the end of it. Yeah, so we can open the question. Thank you.
Hi, thank you for that.
Sure.
I'm interested because we're running the same comparison. And you didn't show any image because well, let's just talk about N. This is an experiment run twice. We have the same N, so we're not willing to make any conclusions based on 2 runs.
Yeah.
The other thing is that you didn't show us images. We saw a lot of probes in the Xenium run elsewhere where there was no tissue, which we didn't see on the Cosmics run. So we were wondering what's the specificity there. And did all of your comparisons come from the cell-assigned dots? Or did you do anything outside the cell segmentation, and did you get any information out of that? And did you do any RNA scope as a validation or a gold standard between them?
Yeah, so as I said, like for the cell segmentation, the very first slides, they were all based on the cell segmentation that is coming out of the instrument, but the ProSeg was actually getting the decoding data and do our own cell segmentation. So these were like our own cell segmentation.
Sorry.
Yeah.
I'm just confused about this because that means that you're using your output as part of your segmenter and then calculating again, right?
So no, we get the transcripts out of the transcripts files.
Right, then
Yeah.
you train a segmenter or get your segmentation out of those transcripts. So you're
Yes.
basically using your results to define a cell, and so you're going to get supposedly a good clustering, or maybe I'm not thinking of it right, but I just think it's odd to use your results as part of your segmentation for
Oh yeah, so the segmentation method is not, it's not a deep learning model that gets like trained on it and then, and then do the test set. So it's not a machine learning algorithm, it's just a probability-based method. Yeah, it is most similar to Baser. If you know, the Baser is based on like where the transcripts are located more. The difference that our method has is that it's also get the DAPI images for the nucleus as well. So it doesn't call like a bunch of transcripts that are somewhere but don't have the nucleus next to it as a cell too. So that's the way. But but I mean, after that, doing the clustering and the UMAPs, that is still unsupervised machine learning. So it shouldn't be affected by the segmentation method, I believe. Yeah. And then the other thing that you mentioned, in terms of the images, yeah, I didn't have any images here, but yeah, the thing that you said, we also saw like for the Xenium having some transcripts outside of the cells, but it is important to see that those are not called cells, so those will be discarded, you know?
Yeah, but, I mean, I'm sorry, but
Yeah.
if I'm doing 1,000 clicks
Yeah.
in situ,
Yeah.
I want to see where all my clicks is. The fact that people are only looking at cell identified for clustering, that's kind of, because it comes from RNA-seq and single cell, but we want to see everything, right?
So, I mean, I believe that it's gonna be the issue with like everything. So even if you run like the VCM, you know, you always have the the RNA leakage, you know, outside. So you only look at the RNA that are like in the perimeter of your cell, and then look at those cells. You know, that's the way that we looked at it. And yeah, the other thing that you mentioned, like concluding based on 2 samples. So it is not just conclusion based on 2 samples per se, because we ran like Xenium for the tumor TMAs, and we ran both Cosmics and Xenium on the skin data as well. So yeah, I didn't bring them here. I have some supplementary slides as well for those. Yeah, if you're interested. Yeah.
Well, thank you so much,
Sure.
Azi, and
Yeah,
thank
thank
you
you.
to all the speakers. We are at time for the session, so I want to let those of you who want to, you know, rush out to the next thing, please do so. But those of us that spoke, and those of you who want to hang out for questions, please do feel free to come up and talk to any of us now. Thank you.
Yeah, sure. Sure, yeah, thank you so much. Yeah.